All right. Hi, everyone. Welcome to this webinar. We're super excited to have so many of you joining from all across the world to learn more about the protein engineering competition that was hosted by Adaptyv that Cradle took part in and ended up winning. Really happy to have you here. I'll start with introducing us so you know who's talking. My name is Elisa de Roos. I'm a co-founder at Cradle, and I'm joined here today by Julian, CEO and co-founder of Adaptyv Bio, Constance, my machine learning engineering colleague who did the work for our participation in the competition, and our co-founder and head of ML, Eli Bixby, who will also jump in to answer questions. And so what we'll take you through today is give you a brief intro to who's Cradle, who's Adaptyv Bio, what did we do for the competition. Constance will then walk you through the Cradle approach and give you a demo of the Cradle platform. We'll talk about the results that we saw during and after the competition and extract some key takeaways. What did we learn from this that is relevant for the field? We'll get to question and answers in the end. Some of you have already submitted questions, which we'll address. But please also feel free to drop your questions in the chat as we're going along, and we'll take those and be able to answer them at the end. So without further ado, I'd love to tell you a little bit more about Cradle. In a nutshell, scientists use Cradle to design better proteins in fewer experiments. We are a software platform that is used to accelerate the journey all the way from the early stages in hit identification through to lead optimization and beyond. So imagine that you are looking to optimize a antibody, its binding affinity, but also expression or solubility or stability. Then what Cradle allows you to do is to bring generative AI into your workflow. So you have the design happening in silico. You can generate sequences using models at Cradle, test these constructs in your lab or a lab of your choice, and then simply upload the data back into the Cradle platform so that the models learn and become an expert at your project and optimize these properties for your molecule, for example, your antibody, as the rounds progress. The neat thing here is that it doesn't require you to have specific machine learning knowledge. And so not everyone like Eli and Constance is a machine learning expert. It's really available to wet lab scientists as well as their computational colleagues. In the lab behind me, this is actually not a green screen, is where Cradle runs wet lab experiments. We do this so that we can A/B test any iteration of our models to make sure that they are robust and have seen real-world data before they're shipped to you as potential customers. We don't actually offer CRO wet lab services. We're also not developing assets in-house. Really see it as the testing grounds that we're using to improve our models continuously, kind of like a self-driving car company would have cars on the road driving through different neighborhoods to make sure that they don't hit a chicken by accident here and there. The business model of Cradle is very simple. So we are a software solution, a SaaS. Everything that you upload into Cradle stays private and secure to you, so your data is never shared between different users. You pay for Cradle on an annual basis for usage of the platform, and any IP that you generate, any new sequences that these models come up with that improve and get you to your project goals belong to you. We're super proud to work with a lot of customers in the field, many in pharma, but also in non-pharma like chemical and food applications. We've run many different programs across different modalities. We're talking about antibodies today, but you can also optimize enzymes, peptides, or other molecules for all sorts of properties. Julian, over to you for an intro to Adaptyv.
Perfect. Thanks a lot. Hey, everyone. My name is Julian. I'm the co-founder and CEO of Adaptyv Bio. So at Adaptyv, we're basically fulfilling the other side of the coin. So we're running wet labs as a service for customers. So you can come with your designed proteins and we will run them in our lab for you at very high speed, very low cost, so that you can get high quality data to improve your machine learning models. Because after all, it's called data science. You need a lot of data. And for AI-driven protein design in particularly, you need fast iteration time. So you can come with your design of new proteins. You can send them to our platform via our website. We will build, synthesize the proteins in our lab. We will then test them for their function. So we have a range of assays that you can choose from, and then you can get high quality data that you can learn from in order to improve your machine learning models or your proteins for your protein design programs. Basically, we're unlocking this like testing bottleneck here, where it's very easy to generate lots of proteins in the computer. Then you have a shiny 3D protein that exists in your computer, but you haven't tested it yet. And we're unblocking this design test bottleneck with our high throughput wet lab. We're running that in Switzerland, and where our company is based in Lausanne. And you can basically choose from a range of different types of data. So let's say binding activity, binding affinity, thermostability, or enzyme activity. Set up your experiment in a very straightforward manner through our website, upload your proteins, and then our lab will generate the data for you, and you'll get notifications whenever like there's a status change in the lab, whenever DNA has arrived, the proteins have finished expressing, and we have generated the data for you. And that's like within like a 3-week turnaround time. So very easy to get started. If you want to learn more afterwards, please reach out or just go to our website at adaptyvbio.com, and you can find out more about how we work with customers. Moving on, we're going to briefly present the protein design competition that we ran at Adaptyv Bio. So the idea here was that we, as a company that generates data, we wanted to show that it gets very approachable now to design proteins. And there will be many people that have cool ideas for new proteins. And we wanted to show that we can actually experimentally validate those proteins so that the AI designs are actually getting somewhere. It's not just proteins that won't express, that won't bind, but actually the AI models are getting good enough that they can make a real world impact, that they can improve therapeutics or design completely novel ones. So we posed a challenge at the end of last year where we basically said, hey, let's design a new binder against this membrane protein receptor, EGFR, epidermal growth factor receptor, which is an important cancer target associated with many types of cancers. And there's a commercial antibody out there that is targeting this, that is being prescribed and basically makes like billions of dollars a year as a therapeutic. And we wanted to see if people could already design better binders for these targets. So basically, participants had 3 weeks to design new proteins. We allowed 10 submissions per team. And we told participants that they had to be at least 10 mutations away from known proteins, right? To have some degree of novelty, that you cannot just change one single amino acid and then call it a new binder. So you had to improve basically upon the existing proteins. And then we validated 400 of those proteins for free in our lab at Adaptyv. And the outcomes were really great because we had over 130 protein designers participate in our second round. We ran the first round before, that was a bit smaller. And then the second round was really where like most of the people had found out about us, and there were like lots of protein designers joining. So we had like 130 participants, over 1,000 novel proteins that have been submitted. And then out of the 400 that we tested, we had over 90% expression rate that we saw. So people really designed some really good expressing proteins, and over 50 basically binders that's bound to this protein, this like growth factor receptor, and basically could have like a therapeutic effect here. And people really created lots of cool and diverse proteins, from improved antibodies, like you can see here, to new nanobodies, to completely novo helix bundles, like alpha helix bundles, de novo beta sheets, this like solenoid protein, mini proteins, proteins that took the natural ligand, EGF, the epidermal growth factor, and mutated that to binders or proteins where people linked different binders together in order to improve basically binding via avidity. So that showed really like the creativity of the community, and that was great to see. For 400 samples, measuring this binding means we had to run a lot of assays in the lab. So briefly walking through the process, what happened is we got these protein designs from all the participants. We then ordered a bunch of DNA, so basically the gene fragments for encoding each one of those proteins. We expressed the proteins. We're using a cell-free expression system for that. And then we ran binding using a technology called BLI, biolayer interferometry, that I'll show a bit in more detail on the next slide. process all the data, and then get the binding affinity out of the basically analysis of the binding curves that shows you how much the binder is binding to the target. So that just like as a snapshot of the complexity of the wet lab site that we're handling with our platform here, that allows the participants to basically just upload protein designs as amino acid sequences and get binding affinity out of the platform. So quick intro to like biolayer interferometry, how we're measuring these like binder target kinetics. The basic principle is we have an optical fiber that we're dipping into the target solution. And on this optical fiber that you can see here, there's capture proteins that capture the designed proteins of the participants. So we immobilize basically the proteins of the participants on this optical fiber. And then based on the reflecting, basically the refractive index change of the optical fiber, we see a shift whenever something attaches to the tip of this fiber. And basically this way we can measure very precisely in a non-labeled way if the proteins are binding to binding targets. So we immobilize the antibodies or the proteins that bind that participants designed. We then dip into the target solution. So you can see here the association of the target to the protein. And then afterwards, we dissociate again in a buffer solution, and the target proteins will dissociate over time. And from this like ratio of association to dissociation, you can basically calculate the dissociation constant, which measures the binding strength of the proteins. And then you can do this for different concentrations and get like very precise measurements of like the binder target kinetics. And for that, just a quick shout out, we're using the Gator Bio BLI devices, which are really great. Can highly recommend running this. And basically with this, we ran many replicates for all of these samples and generated the binding kinetics that will show you on the coming slides and like the results from this competition. So with this, I can show quickly like about Cradle's approach, and Constance will give a demo afterwards. So I'll hand it over to you.
Yep. Thanks, Julian, for giving an overview of the competition. So we'll dive right into Cradle's strategy. So to give you the full story, we had just signed up for NeurIPS, and my colleagues and I were kind of looking at the relevant workshops, and we found the AI DrugX workshop where Adaptyv was hosting their second protein design competition. So naturally, this seemed like a good competition for Cradle to participate in, which is why we sat down and we initiated the generation. So to initiate a generation at Cradle, we can do this in a few steps. All we need is a lead sequence. So this is a starting point from which we can generate mutants. If we want to block any positions, we can do that. If we have any experimental data, we can upload it to improve our predictors and generators. And we can also set constraints on the amount of mutations we want to see in our mutants. And then all you have to do is click generate. So I'll actually give a live demo. This really took us, or at least took from my time, a few minutes. So we'll do this together. Okay. Okay, so you should be able to see now the Cradle's platform. So here we're in the demo project. Like every one of our customers have their own workspace. We can create a new project. So this would be EGFR binder. And then here you have to paste your wild type or the starting sequence. So in this case, we use an SCFV reformatted Cetuximab. So the sequence I just pasted, and we can create our project. So now we're within our project, EGFR binder. And to get started, we want to add project assays. So here, what do we want to optimize? We want to optimize for binding affinity. So the task is to optimize binding. Specifically, we want to optimize KD, and the unit is in nanomolars. We can also add other assays. In this case, we just have binding affinity, so we can continue. Here we can import experimental data. So if we had any experimental data, we would upload it. Otherwise, now in this case, we didn't, so we skip the upload. And then we can set our objectives. So here we have binding affinity, which we want to decrease the concentration of, so to increase binding affinity. So we can add this. If we had other assays we were taking into account, we could also add constraints to these objectives. But for now, we'll save and continue. Here you can see that if for some reason we had experimental data later, we could still upload it to the platform. Our project assays and our objectives are completed, and so we can start our 0-shot round. So Here, I'll create the round. Now we're in our round 1. This is a 0-shot round, and we can configure our generation. So for this, how many candidates do we want to generate for the competition? We initially generated 10, so I'd set that to 10. The starting point would be the scFv reformatted Cetuximab, so the project-based sequence. Then here you can set the number of mutations. So for the competition, we know that we needed a minimum of 10 mutations, so we set the minimum to 10. And we let the maximum be automatic so that our models could best decide how many mutations to include. And then we also made the strategic choice to block CDR regions within this scFv reformatted Cetuximab. So in this case here, you can add the block mutation. So here we want to block all mutations on, so we're going to define a new region. We're going to define the CDRs region. Here you can include your CDRs by like adding them manually like this, or for this purpose, I'll make it easier. I'll just paste all the positions. So these are the positions that we're blocking. So you can save this. You can also add other positions if for some reason there's another region that you'd want to block, but here we're blocking all mutations on CDRs. Done. And now we've added all the information for our generation. Maybe I'll write down like this is for the protein design competition. And we can start generating our sequences. Now if I refresh this, you can see here we have our sequence generation that's running. Now this part, this runs on the cluster for a couple hours. We're not going to wait for this to be done, but what I'll do is I'll skip directly to the actual output of this. So once generation is done, you get a nice generation report. So this is the generation report for the 12 sequences that we tested in total towards the end. So we originally generated 10 and then we generated 2 additional ones. So here you can see that 12 sequences were chosen out of 800,000 that were screened. We get other stats about this. But the most important is here we have our generated candidates. You can see the mutations that are included, the sequence that was generated, the template it was generated from. So this generated sequence includes mutations on this template sequence, other information, and then a performance score. And this is a score we get from our models. You can also take a look at the 3D structure. Here in green, you have the positions that we blocked, and in blue, we have regions where our models found promising mutations. So you can take a look at this. And then we also have a breakdown of the mutation frequency. So here you can see, for example, that at position 154, this mutation was quite frequent. Other mutations were less frequent. Again, we let our models decide and guide this choice. And we can also see that from the 12 sequences that we designed, they all have 10 mutations. And this is the output you would get that you would analyze before sending the sequences off. No. Yeah. So once we reviewed the generation report, we looked at the final sequences that were generated. We had 12 sequences, or in the first case, 10 sequences, 10 mutations per sequence, with 25 distinct positions and 33 unique mutations at those positions. And once we were happy with this, we sent them to Adaptyv. And we can see that our models recommended a diverse set of sequences. So Cradle, rather than designing one sequence at a time, we designed sets of sequences to maximize the chance of a successful round. The idea is not to put all your eggs in the same basket. So although we designed 12 sequences that were exactly 10 mutations away from the same lead sequence, some of that we noticed that in our set of sequences, some sequences are more similar to each other than others. Specifically in this heat map, we have the self-alignment scores minus the Blossom 62 substitution scores. So in this case, higher scores means mutations that are less conserved, which could mean that there are more functional or structural differences. So we can see that across the 12 sequences, we have a varied set of some sequences that are more similar to each other than others. So now we can look at the results of the competition. So Adaptyv received about 1,000 sequences, and initially they ranked all these sequences with some in silico metrics. So 2 metrics from AlphaFold 2, IPAE and IPTM, and then also the ESM2 pseudolog likelihood scores. So initially, Cradle's design scored quite poorly on these metrics, and only none of them were selected for lab validation from the top 100 designs. Luckily, Adaptyv handpicked 300 additional designs, from which one of them was our design. And this design actually went on to win the competition. So in the competition, we see that there are two designs that significantly outperformed the SCFE reformatted Cetuximab, and our design outperformed the nearest competitor with a fourfold improvement. Now, after winning this competition, Adaptyv gave us 11 free assays for us to test more sequences. So we decided to test the 9 remaining sequences that we initially generated, and then we generated 2 additional sequences in the same way. And what we found is that all 12 mutants would have won the competition. They would have placed top 12. So in this plot, we corrected for batch normalization, and we identified that 3 out of the 12 sequences were sub-nanomolar binders, and then these would have outperformed actually the competition winner. And all these sequences outperform Cetuximab and the other sequences from the competition. Now, although these results are very promising, a natural question is, did we maybe negatively affect another property in the process? Maybe we improve binding affinity at the cost of expression or other properties of interest. So when diversifying a lead binder, we don't have direct control over the effect of the properties of interest. So what we did is that we used MHC2 binding predictions to assess the risk of immunogenicity, which is one of the main properties of interest when designing a binder. In particular, here we see that a higher number of MHC2 binding peptides in a sequence, because it indicates a greater potential for the presence of T-cell epitopes. This would maybe lead to an unwanted immunogenic response. So given an input sequence, and net MHC2 predicts the binding affinity of each peptide within the sequence to different alleles. So here you can see that the sequences we designed don't indicate that we bind to more of these alleles, which would indicate that we didn't introduce any unwanted immunogenic response. Obviously, these are predictions, and it's possible that beyond this, we could have introduced an unwanted immunogenic response, but at least for the moment, the predictions don't indicate that we did. So finally, we can talk about the main takeaways of this competition and our results. I think first, something that the community, I think, has already pointed out, and that Adaptyv has mentioned in one of their blogs, is that in silico metrics don't tell the full story. Here we see the correlation between the in silico metrics that Adaptyv used to rank the sequences at the beginning of the competition versus binding affinity. And what we can see is that there are very poor correlations between these metrics and binding affinity. And there are actually going to be more insights in an upcoming paper that Adaptyv is writing. And then, so beyond just the correlation between binding affinity and these metrics, Adaptyv also looked at, could these metrics at least tell us whether or not the sequences are binders or not, or if the sequences would express or not. So whether or not these metrics could be used as binary labels. And the short answer is no. So when plotting the ROC curves and computing the area under the curve, we see that most values range in the 0.5 region. And this basically means that these metrics are not better than a random classifier. So again, this is these metrics can't tell us better if a sequences will bind or not bind, or express or not express better than a coin flip. Another important point is that for affinity maturation, lead optimization seems to be more successful than de novo design. De novo design was the most common method for generating sequences in this competition, and we hear a lot about de novo designs these days. But in fact, only 4 out of the top 100 binders were from de novo strategies. So this highlights that de novo it remains a very difficult, unsolved problem. However, this is a good moment also to talk about the difference between de novo and lead optimization. These are two different problems. Usually lead optimization actually comes after de novo. So generally, when you're designing a therapeutic, for example, the first step would be to generate your protein. So please generate a binder to this epitope. In this case, please generate a binder to EGFR. And then once you have some binder to start with, this could be a weak binder, you enter the phase of lead optimization, where you may want to include more properties. So for example, expression, stability, and binding affinity. And here, the idea is please optimize this set of properties from this lead molecule. And this would happen round over round. Specifically at Cradle, this would happen round over round with machine learning guided designs. So specifically for this competition, the designs we generated were in this first part of lead optimization, where we don't have any experimental data, which is why we call it zero shot. But it is still part of this lead optimization where we optimize a known binder. So these are distinct problems with varying levels of difficulty, but also trying to solve different problems. And finally, something we've seen in the community and that we strongly believe is important at Cradle is the question of diversity. So there are two independent goals of diversity within this problem. One is more on the design side. It's how do you minimize downstream experimental risk? So you want to have diversity in the set of sequences that you're designing and sending to the lab. Specifically for zero shot, we want to diversify through mutational overlap, or as we talked about before, looking at Blossom 62 scores, for example. So the idea is, in this first step, we just want to generate sequences that are diverse enough to get information about different combinations of mutations. And then when we enter the multi-shot phase, where we have experimental data for the properties of interest, we want to use these functional landscapes that we learn from this experimental data to sample meaningfully diverse variants. And by meaningfully diverse, we mean with respect to the multi-property optimization problem that you're solving. And then there's another independent goal of where diversity comes into play is how do you diversify from existing IP? And this is something that at Cradle, we don't try to answer specifically. This is something that we expect our customers to define, whether or not this is in terms of number of mutations or something else. And this is also to minimize the commercial risk. So for this specific competition, this was more on Adaptyv side of the fence, where for them, something that was diverse enough was a binder that was 10 mutations away from a known binder. So these are two different goals that we have with respect to diversity. So finally, just to recap, what we've shown here at Cradle is that Cradle's zero-shot diversification pipeline is effective at exploring headroom beyond initial lead. So in this case is that we found that there was headroom in optimizing the framework region of an scFv reformatted Cetuximab. The next thing is we've seen that off-the-shelf in silico metrics can cause you to miss winners. Initially, only one of our sequences was chosen and it won the competition, but if all of our 10 sequences were chosen, they would have won top 10 in the competition. So this calls for better in silico metrics and also believing in your strategy. I think for us, it's once we saw that our sequences didn't rank well, we decided to leave it as is because we built a whole technology around this. So we trusted our designs. And then for what we continue to see is that for affinity maturation, lead optimization, so optimizing a known sequence continues to be a more effective strategy than the de novo design. Although here we do point out that de novo and optimizing from a lead are different problems. And then finally, one point we'd like to make is that we need clearer lines around diversity, novelty, and patentability. Legally, this isn't even a question that has a clear answer, but it's something that has come up in this competition. Thank you so much for listening. One thing that we'd like to point out is that we're going to be publishing our 12 designs soon. So if you want to have access to the sequences, take a look at them. We'll be doing that really soon. So stay tuned, and thank you for listening.
Cool. Thank you, Constance and Julian, for elaborating. We will take some questions. Many questions in the chat. Some of them have been answered. I'll try and kind of group by category. Constance, can you say a little bit more about the design approach and like why did we choose to block the CDRs? Did we just get lucky, or is that something that is a reliable strategy? You're muted.
Sorry, I didn't realize that I pressed the mute button. Yes, so why did we choose to block the CDR regions and make framework-only mutations? So the real story around this is that right before this competition, we had already ran an in-house project where we made framework mutations, and these had shown to be quite successful. And we had some suspicion that it was because there was headroom in making framework mutations, and making mutations directly in the CDR regions, which are regions that are directly responsible for binding, obviously can be a bit riskier. And since this in-house project had led to successful results, we decided to test it again through this competition. And then once we got the results back from this competition, this kind of confirmed that this wasn't luck, and it's just we're able to find headroom in the framework area. But the main, I think the main explanation for this is CDR regions are directly responsible for binding, and so if we're starting from a known binder, we know that this binder already binds to EGFR in this case, then by making framework mutations, you're making smaller changes, kind of like you're just changing a little bit the orientation, so maybe it'll come and bind more closely, but you're not directly changing the part that's responsible for binding.
Cool, thanks for that. A question for you, Julian. Can you comment on how accurate the K-on and the K-off rates were that you found? And is this a standard assay that you ran, or do you kind of optimize assays for different properties?
Yeah, so this was our standard binding assay that we've run across like thousands of sequences, many dozens of customers at this point, and different targets. We have like a pretty standardized assay setup here that I just explained in the chat to one of the participants. Basically, if you're asking like how good is the data quality, you can ask, for example, can you measure a wide range of KDs? So that we've shown, for example, we can measure from like low nanomolar to picomolar binders up to like micromolars, basically very weak binders. So that you can see in the data. And basically, then the other thing would be how is the basically assay replicate reproducibility. And for that also, we get like very good within like basically typically sub half an order of magnitude deviation between different assay runs. And typically for like these kind of kinetic assays, 1 order of magnitude is typically like the noise level that people see often, and we're much below that. So you can check out all the data actually on our GitHub, and we have all the raw data included there as well, and you can basically download this and run your own analysis. But the data quality is really good.
Cool. Then a question for you, Constance, that came in ahead of the webinar is, what do you think makes Cradle successful in protein design competitions? Is it the quality of the training data, or is it something unique about the model architecture tailored to this particular problem?
Yes. So first, to answer the part about the training data. So in this specific case, we didn't have any experimental training data. So we used our zero-shot diversification pipeline. So that's the first step of our ML-guided lead optimization pipeline. And the idea here is we want to find headroom, and we want to generate sequences that we believe are going to be stable. And we're not optimizing directly for a property. So in this case, like we weren't actually directly optimizing for binding affinity. We didn't know if the binding affinity would be strong enough, but what we can see is that we designed 12 sequences, and out of the 12 sequences, 12 of them were binders, so we're able to find good leads. And in our zero-shot diversification set up, I think what we're good at is diversifying and not putting all our eggs in the basket. So maybe if there was a bit less headroom, we wouldn't have gotten 12 out of 12 binders, but we still would have gotten a high percentage of binders because we allow for this diverse selection of sequences. We select a set of sequences, not one sequence at a time. Yeah. I think that's what makes us strong, yeah.
Thanks. Then there was a question on how you went about, or how the models went about down selecting from the 800,000 sequences that were generated. Like, how did the Cradle models decide which 12 to send to the lab?
Yes.
And maybe you can wrap in like how to understand the performance score.
Yes. So that performance score that I showed on the platform, if I'm not mistaken, are log likelihood scores. So these are scores that we get from our generator, or our set of generators. So the way that we down select is, so as I said before, we don't select one sequence at a time. We're going to build a set, and we have certain constraints on that set. So the minimum number of mutations, the fact that we don't want to see the same we don't always want to see the same mutations appear in that final set. The way that we diversify in zero shot is by not allowing certain mutations to appear too many times. So what we'll do is we'll start by choosing one strong sequence that has a good score, and then we'll little by little add sequences to that set that match our requirements. So the requirements is, if you get a second sequence that's very similar to that first sequence and also has a high score, maybe you won't initially include it, because you first want to find another sequence that also has a high score that's more diverse from that initial sequence that you added in your set. So we have this trade-off between diversity and high-performing sequences.
And so maybe a follow-up question to that is, with the in silico metrics that were used, right, 11 out of the 12 designs would have actually been overlooked. And so how do you kind of balance the cost and time that it takes to generate data on the one side, and then the fear of missing out on potentially winning designs on the other side?
Yeah. So again, this kind of plays it's the same kind of answer where you want to include diversity in the sets of sequences that you're generating. Like, yes, the final goal is to get hits, and you want to get the more hits, the better. But the thing is, if your model is telling you, okay, these few mutations are going to give you good results, and you only include sequences that include these mutations, you're taking a lot of risk, because if the model is wrong about this, then you've just designed a plate of 96 or I don't know how many sequences that are not going to be hits. Whereas if you balance out where your model finds promising mutations, and where and diversity, if you balance these two out, then when you get the experimental data back, you're going to see that some of these sequences are good hits, others maybe not, but at least your model learns what is a good hit, what is not a good hit. And this is even more important, I think, actually, in the multi-shot rounds later on, where we do start getting experimental data back, which is that negative data is always important and good data. So even if you design sequences that actually don't end up really matching your constraints, that's still really good information for your models, and then you're going to get even better models for the next round. So yeah, this is why, again, the way that we generate sequences is we don't see it as generating one sequence at a time. We see it as generating a plate of sequences.
Cool. Then there was a question, to what extent is this the result of Cetuximab or EGFR being a well-studied lead, presumably represented in public training data? Is this driving the success? Or put it in another way, how successful do you expect to be with a lead on a novel or a very different target? And has Cradle done any benchmarking on this?
Yes. So in terms of this being a well-studied protein and data, and so there's data on this protein out there, I don't think that has had that much of an impact, because we didn't go and look for that data, and we didn't use it to train our models. the fact that it's a well-studied protein is that maybe we start, like, the starting point was already a really strong binder, and so naturally, like, if you start somewhere higher, then like you're gonna design sequences that are better. That said, I think it also has a lot to do with the headroom. If this was a protein that didn't have that much headroom, then maybe we wouldn't have found as many hits within our 12 sequences. And I think that this depends this depends on different projects, the different starting points. It depends if there is headroom around or not. And I do think that this is something that you can maybe see with models, especially when you start getting experimental data back. You can see when your models kind of hit a wall, when they're not able to find novel mutations that you haven't tested. Or maybe like your models start noticing that you've explored all the potential novel mutations, and then maybe the sequence, the target that you're looking for maybe doesn't exist. And we've definitely seen that case before where we've hit walls. And in terms of optimizing, maybe Eli, you know better, like in terms of optimizing, like from a zero shot round, maybe you know better. I don't know.
Yeah, I mean, we have seen in cases, for example, in super chimeric proteins, where the chimera itself is not well represented in the training data. We have seen you can correct for that by sort of getting the parent sequences, and finding related sequences based on the parents, and in that way you can kind of fix this for zero shot approaches where there is some design. We haven't yet answered the question for like truly de novo sequences that aren't represented at all in the training data. How does this work? We have an active collaboration, which I can say very little about at this point to answer that question. But that's very much an open research question of where we really don't have good data coverage in sequence space. How well do these approaches work?
Then another question that came in is Jen asked, do you have similar public examples of protein designs that involved the iterative cycles of testing and training? Because this is an impressive zero shot, but most use cases actually require additional optimization.
Well, the beginning of the question was, are there any public?
Yes. So multi-round projects where Cradle did well.
Yeah. I think we have a blog post on, if I'm not mistaken, a blog post on one of our projects where we did multi-property optimization, right?
Yep. The
Yes.
Align to Innovate blog post, Align to Innovate protein engineering competition, was a data set with different enzymes and different properties over multiple rounds, and that can be found on our blog post at cradle.bio.
I think that pretty nicely covers the sort of predictor and multi-property predictor building portion of doing iterative optimization and this kind of lab in the loop design. I think what we don't yet have a ton of public case studies for, but I think we have some in the works. And we certainly run a lot internally is covering the generative portion of the loop. How well do you do in generation in the second round after you've seen lab data? And I think this is actually one of the strengths of our technology, is that we're able to actually condition the generator as well as building predictors. And you do see that a little bit in the Align to Innovate post as well, because we report the correlation of these log likelihoods we use to build to generate the sequences. So earlier, Constance was talking about that's what the performance score is here. We correlate those with the conditioned generators and the measured properties, and see that even when you treat the generators as predictors, they perform quite well. Obviously, the best proof of any generative system is in like a forward-looking generation where we actually go and test the experiments. And so looking into publishing some of those both from our internal lab and partners.
All right. And then Alejandro asked, whether you also tried predicting other developability metrics other than immunogenicity, like polyreactivity, aggregation, propensity, thermostability. And maybe this links to another question that was also asked, like which properties does Cradle support, and what are are there any that are out of bounds, or when is a property out of bounds?
Yeah. So to answer the other developability assay portion of this is no, we didn't look into other. So I think we looked into humanness a bit. We used a humanness predictor, but other than that, we didn't. This is mainly because these assays are very complicated and expensive to compute. But I think like it would be really cool if we could take this project further, and I think maybe we're like starting to look into this. And basically for an assay to be used by Cradle, what we need is we need it to be an assay that you can a property that you can assay in the lab for a plate of sequences. We need at least 100 sequences approximately, or like 100 data points for it to be useful for our algorithms. So in this case, like it can't be a low throughput assay, and I know that can be the case for some of these developability assays. So that's kind of a limitation that we have. But yeah, so then to answer the question of like what does Cradle support, we basically support any assay as long as you can give us at least 100 data points from that assay. And we also, I guess, another requirement is we need this assay data to be quite clean, for it not to be too noisy, so that our models can actually learn. This is something that we can benchmark and we can check before we actually start generating. So yeah.
All right. And in terms of how many mutations were introduced, of course, this competition set 10 mutations as the lower bound. How would a user of the Cradle platform go about choosing the number of mutations, setting a minimum or a maximum?
Mm-hmm. I think we always offer defaults. Like I know for zero shot, usually the default is 1 to 4 mutations, although we do have customers who set it to much higher. We had this very recently, actually. But this is so this is something where you can use the defaults from the platform, or you can set them yourself. Again, this is something, I think this is more of like a question the customer has to answer. Depends like if they have any constraints on their side in terms of, for example, IP. But for Cradle's algorithms, this is a parameter for us. So the customer can decide to set it or not to set it. And I think what we usually see is that, so for zero shot, it's usually 1 to 4 mutations. And then later on for multi-shot, maybe when we introduce novel mutations, so these are mutations maybe that we haven't tested in previous rounds, there we'll introduce them in sequences with less number of mutations. And then what we also do is we also do recombinations of mutations. So once the model finds successful, or once we've found successful mutations, we try and recombine them. So this is where it leads to sequences maybe with more mutations.
Looking at the other questions, there are questions like, what if you pulled mutations from a multiple sequence alignment, or you ran ESM2? How different would the mutations be that you got from those approaches versus the Cradle pipeline?
So we actually use MSA, so multiple sequence alignments, as training data for some of our models. So I guess implicitly, like, it's not that we directly pulled them from the MSA, but we definitely use like our models learn information from these MSAs. So which mutations are conserved and not conserved, this kind of information is included in our training data for our generators. So implicitly, I think this gets this is what like when our models suggest promising mutations. I think this.
I think it's also worth pointing out that kind of the grab sequences from the MSA just kind of begs the question, which MSA? And I think one of the things in focusing these models on our particular region of sequence space, one of the things we put a lot of work into, and actually this was some of the earliest work that Constance did at Cradle, is identifying sort of what the right level of variability is and what the right search tools are, et cetera, for building that initial training data set. And that's something that's done completely automated. So it's not that you have to go out and find your own MSA that is used as input to the training data process. That would be accomplished in the clicks that Constance showed you earlier.
Maybe a final question is, how can you get started? Do you need to know an epitope? What if you have a totally novel target? What if you have no lab?
So for the no lab part, at Cradle, so as you can see behind Elisa, actually we do have a lab, but this is only for internal purposes to be able to optimize our algorithms internally. But if you don't have a lab, you can always turn towards CROs. For example, Adaptyv, you could generate sequences with Cradle and then send them to Adaptyv for Adaptyv to assay them. I guess there are other options, but yes, this is a requirement that we, at least at Cradle, have for our customers, is that they need to provide the experimental data for us. Our internal lab has a different purpose here. There is a list of things that you said, Elisa, and I can't remember the rest.
What if? How much do you need to know about the target or the epitope? And can you start with just a target?
So you can't yet start with just a target, but what you can start with is screening data where you've not yet identified a target. And so this is basically the basis, or not yet identified a hit. Sorry, screening data against a target where you've not yet identified a hit. So this might be panning data from a phage display campaign or fax data. And what we've demonstrated now a few times in-house, and we are hopefully we'll publish about soon, is that we're able to identify hits within this set and then actually diversify them like we did here in that initial round along with the identification. So not yet, full de novo, really focusing first on the later stages of the optimization pipeline, lead optimization and hit identification. But yeah, we do feed that NGS data directly into our models and use that in initial rounds.
All right. And then to round it off, maybe some perspective, like, will we get to a world in which no wet lab work is longer needed and Julian and Adaptyv go out of business?
Yeah.
I think
if I can
yeah.
Julian, do you want to answer that?
Basically, yeah, my just like 2 cents on that is basically the frontier shifts, basically, right? The more complicated things, you always need new data. So if you don't have a feedback loop with reality, you're always limited to what you've already seen. And I think the basically set of problems in biology is basically infinite. So until we all live forever and can atomically precise manufacture any material, there are basically problems in biology to solve.
I see a lot of nodding from both Eli and Constance.
Yeah, I think the ML viewpoint on that is completely the same, but the phrasing is just flipped, which is every model eventually fails to generalize, right? And so you just expand that trust region, you expand that generalization window, right? And then you travel to it, and then you still need data beyond that generalization window, And so we will see that generalization window expand, but we'll still need data to push that farther.
All right. I think with that, we will round out. Thanks, everyone who listened in. Thank you, Julian. Thank you, Constance and Eli. And we hope to keep you tuned of news to come out of both Adaptyv and Cradle Labs in the future.
Thanks for having me. Thanks for joining, everyone. Bye-bye.
Thanks for attending.
Thank you.
Thank you.
Bye.